Everyone. My name is Sarvar Singh and I am from IIT Jodhpur. My today's talk is about trace level molecular detection in organic honey that is relevant for therapeutic applications. So, as we all know, the honey is basically a sweet viscous liquid produced by honey bees. So basically, it contains carbohydrates and fructose, maltose, maybe flavonoids, amino acids, and other components. And it has been used in therapeutic applications since thousands of years, not only in India but in all over all the other parts of the globe as well. It has been used in Indian system of medicine, in Greek system of medicine, in Egyptian system. You might be surprised that it was cited 900 times in different drugs. So and in Ayurveda, as we know that whatever we give with honey, then it enhances its properties multifold. So well, uh, these are some of the properties. It may have antibacterial, antiviral, and antioxidant properties. But what are the molecules responsible for it? And the honey which we are using, whether it has those molecules or not. So that is one of the important aspects of this talk. So my focus will be on organic honey and its contamination detection. So if there is any contamination, the honey, we cannot term it organic. So it is the honey which has higher nutritional value, which has no contaminants like pesticides, insecticides, or antibiotics. So there can be multiple ways which can be direct or indirect, which can turn up into the contamination of the honey, such as adding the sugar syrups by the bee farmers or feeding the bees with sugar syrups and then producing the honey might also end up in higher quantity of the sucrose and the artificial sugars in the honey. And maybe by for commercial purpose, the bee farmers might uh, contaminate or adulterate it with different type of sweeteners. And if they are treating the honeybees with different antibiotics, that might also end up in trace level in the honey, and which may be responsible for antibiotic resistance, because whatever is less than therapeutic dose can be harmful for the body. So these are some of the mechanisms through which the honey can be contaminated. But why the trace level molecular detection in honey is important? Because currently the techniques which are being used are, first of all, FSSAI is mute about the additional sweetening of the honey. It is when we search about pesticides, or we search about honey, then the only thing comes is the antibiotic. So if we search the FSSAI food standard and um, safety regulations 2021, if we search the keyword honey, then we find that it only talks about antibiotics and pesticides. But so all kinds of honeys with additional sugar and syrups are are available in the market. But the European standards and the American standards are very much stringent than Indian standards. So uh, it also talks about the which techniques for the antibiotic detection we can use. So it says that liquid chromatography, mass spectroscopy, and NMR spectroscopy can be used for the antibiotic detection, but these are very expensive, labor extensive, and also time consuming. Another thing is that we can only do the testing in a laboratory. So Raman spectroscopy is what we are exploring, because it can be used in a portable manner and at the point of use. So but it also suffers from one limitation, that is that the signal from conventional Raman spectroscopy is quite low, and we need to enhance the signal somehow, and that is why we are using surface enhanced Raman scattering, and we are using some metal nanoparticles deposited on the surface. And as sir was talking earlier in one of the slides, that if the molecule goes into those hotspots, then we might get the enhanced signal. So we are trying to engineer those nano gaps and those nanoparticles, and we have fabricated, this is the experimental setup, the schematic of the experimental setup. Let me briefly talk about what is Raman spectroscopy. So there is a laser source which we shine upon the sample, and the light gets scattered. The inelastically scattered light goes back to the detector, and it has information about the molecule on which we are shining the light. So we get some peaks, and those peaks represent the molecular vibrations. And with the combination of those peaks, we can say with certainty and with high specificity that this is the molecule which is present. But in search, this signal enhances 10 to the power 7 times, 8 times. So this enables the trace level molecular detection also, which is otherwise not possible in the conventional Raman spectroscopy. So we have developed this substrate. So these are the pictures from the different stages of the substrate development. So what happens is that whenever the molecule goes into these gaps, so the first picture represents the silver nanoparticles deposited on the silicon surface, and later we are creating holes in the surface just to make it random and uneven surface, and further we coat it again with different size of nanoparticles, because we know that what will be the appropriate and the most optimum size and the gap for the molecule, so that we can get an enhanced spectra. So the second thing after developing it was that whether the chip is working or not. So we tested it with a model molecule, that is rhodamine B, and you can see uh, in this one that the first line, which is almost no spectra, was coming from the bare silicon surface. But when we increase the molecular concentration of rhodamine B, we got some signal, but it was again on the bare silicon, and it is around 50 molar concentration. But why we say trace level molecular detection? Because this signal is coming from 1 micromolar concentration of rhodamine B. So there is around 10 to the power 7 times enhancement in the signal, just because of these gaps. So, and another interesting thing is that we also got the uniform enhancement all over the surface. This picture is from the different spectra of 150 different spots. So everywhere we were getting enhanced signal. So 1 micromolar concentration, anywhere you shine the laser and you get a signal. So what more interesting can be there? So this technique we used for actually for testing the honey samples. So we took two different types of honeys. One was organic honey; it was a certified organic honey, and the another one was a local market bought honey. So we wanted to see what are the molecular difference. Is there any difference if we can spot from generating the signal using our nanosensor? So we observed that there is a very clear cut difference between both the honey sample spectra. And additionally, since FSSAI is emphasizing more on the antibiotic presence in honey, so we also thought of taking one antibiotic, that is chloramphenicol. We observed its spectra and we tried to match it with the honey spectra which we got from two samples, and so that if there is the presence of chloramphenicol in the honey samples, we will be getting some signal from these honey samples also. So you can see here that these two peaks, which are 608 and 771, these are specific to sucrose, and these two peaks are absent in the organic honey. The H1 represents the organic honey, whereas the another difference which we could spot was that from 800 to 1200 range of the Raman shift, we we saw that there are some additional peaks which are certainly not present in the ordinary honey. So we tried to match it with the literature, and we found that most of the flavonoids are having Raman spectral peaks in this range. And well, this range is almost similar, but we have also tried to assign the Raman peaks to different molecules. So this is that assignment. So the difference between both the honey samples is listed here through the Raman peaks. So these are the vibrational identities. So as I talked about that, these peaks represent the molecular vibrations, and with the combination of the vibrations, we can say with certainty that this molecule is present. So these two peaks, 608 and 771, these are only specific to sucrose, but this 754, which is ring vibration, is also present in glucose and fructose. So we are assuming that this is largely coming from these two sugars. and the peaks from chloramphenicol were not matching with any of the honey spectra. So we concluded that the chloramphenicol was not present in both the honey samples, at least in 1 micromolar concentration, because that's the uh, concentration which we observed for rhodamine B. Well, additional peaks are also present, which can represent flavonoids or the other nutritional compounds. So, see, flavonoids is one of the phytochemicals which are known to be bioactive and which inhibit the growth of the various bacteria and viruses. So, if we are using the honey for therapeutic purpose, then we would conclude that organic honey would be a better option. So these are the conclusions of the work which we had done. So low cost substrates on based on optical nanosensors. So we had developed and tested. So protocol for honey testing. Why Because honey is a very thick and viscous liquid, and it was a tedious task to generate the signal because the hotspot is very small in nanometers dimension, and there are two primary requirements. One is that the molecule should go in the hotspots. Another requirement is that the laser should shine at the hotspot, and we should be able to focus on that hotspot between those gaps. So we found that reducing the concentration of honey sample in water, if we are diluting it in 10 nanograms per ml, we are getting enhanced signal, because then there is a high probability that a very few layers of the molecules of honey will be honey components will be there on the hotspot surface, and will be get our laser will be shining on actually on the hotspot. And the third conclusion, we found that sugar content was not observed in organic honey samples, because either the sugar specific peaks were not there, or if the other peaks which were fructose and glucose, they were also not having that much intensity. Ordinary honey was found to be adulterated with artificial sweeteners, as I talked about. No sugar, sucrose content, as I already told. Antibiotic presence not detected. Organic honey was found to have additional molecular Raman peaks, which may correspond to. We have not assigned the Raman bands, but this is what we have observed. And acknowledgements. I acknowledge the Jodhpur City Knowledge and Innovation Cluster, as well as the Center of Excellence for Biotech, Ministry of Ayush, for funding this work, and my lab members of research and innovation in multidisciplinary sensor. This is the name which we have given, RIMS group, and the group leader, Professor Ajay Agarwal. This is the picture of our group. In the chair, the group leader is there, and I am on the extreme right. Well, that's all about my time. Thank you.